Good morning. Good afternoon, students. How are you? All of you can hear me? Hello? Hello? All of you can hear me? Just reply me through chat, please. Okay, all right. Umar Yasir, you can listen me. Okay, all right. So today we are going to discuss about a new paper, okay? Umar Yasir? Umar Yasir?
Yes.
Can you hear me?
Yes, I do.
And the screen is okay? You just check the screen. There is a slides. There is?
Yes, yes.
Huh?
Yeah.
So it means
Yes.
everything is okay, right?
Yeah.
All right. Dear students, today we are going to discuss about paper biology IGCSE 0610 March 2016, paper 42, right? So the first question is, a researcher used a light microscope to observe epithelial cell from a human chicken. The figure 1.1 is a photograph that the researcher made of these cells. Okay, he actually isolated epithelial cell from human chick, and now he want to see, like he studied that. Okay, so different parts of that cell you can see here in the figure 1.1. Now they will ask a question from us, so definitely before asking the question we should know about general thing about these cells. You know, these cells are isolated from human, right? So if these cells are isolated from human, if these cells are isolated from human, so you can, like, just these cells are isolated from human, so definitely these cells, the outermost boundary of these cells should have what? The outermost boundary of these cells are uh, cell membrane, okay? While this part is called what? This part is cytoplasm, while this part is nucleus, okay? And the cytoplasm, there will be different organelles, right? Now if you see here, these cells are irregular in shape, like they having no such a specific shape. All these cells are irregular, right? Okay, now let's move toward the questions. First question is, name the part label A and B, right? Name the parts label A and B. So what I expect from you, please? What should be A? Definitely it's the, like, it's the region between, what? It's the region between cell membrane and nuclear membrane, so it should be cytoplasm, right? While the B part with us is what? It is nucleus, and nucleus contain what? It's contain, a nucleus contain DNA, right? Okay, all right. Now, state one of the function of the cell membrane. So what you expect? What should be that? Like, what should be that function of cell membrane? So definitely cell membrane is partially permeable membrane. It's control in and out of the material, like it allows those material to enter and it allows those material to go out from the cell. So it's actually control the in and out of the material through the cell, right? Second one, like if we know, like there is different channels in the cell membrane through which material enters. So it's contain like certain channels through which different material can enter. Also, the cell membrane contain different uh, antigens for binding of hormone, for binding of enzyme. So all these are different functions of cell membrane, right? Okay, so the first answer with us is cytoplasm. The second one is nucleus, while state one of the function of the cell membrane, so control substance movement in and out of the cell. Other extra I already discussed with you. If you want to study the question, you can. All right, now let's move toward another question. State how the shape of the cell that shown in figure 1.1 different from the shape of palisade mesopel cell and leaf, right? If you see here, like if you look toward this is the structure of leaf. So this is upper epidermis, this is lower epidermis, this is spongy mesopel, and this is palisade mesopel. So from the structure, from the figure, we can, like from the figure we will give the reason, like what are the structure between mesophilic cell and palisade mesophilic cell. If you see here, the epithelial cell, these cells are irregular in shape. These cells are irregular in shape, right? While the cells of palisade mesopels, these are cylindrical in shape. Okay, these are irregular shape. You can also give these are round in shape, while the palisade mesopel, these are cylindrical in shape. Okay, right? So okay, so from here we will just look like we will just look to the figure, and from the figure we will give our answer. So like you can, like if we go to journal, we can also, so like they're like here in the plant cell there is cell wall, but in the figure, like the one cell that given to us, there is no cell wall. But here we will just give our answer from the physical, what is now in our screen. We will give the answer from that. Okay, now okay, is it clear? Now come to our another question. Figure 1.2 show an electron micrograph of a mitochondria. Mitochondria have two membrane, like it's double membrane structure. The one membrane is outer membrane, right? And like inner membrane, the one is inner membrane, while the another is outer membrane. So the inner membrane is folded and refolded. If you see here, so this inner membrane is folded and refolded, like this structure is there. You can see this is outer membrane is smooth, while the inner membrane that is in the mitochondria, this is folded and refolded. Suggest why the inner membrane of mitochondria is folded. Now it is a question. Like mitochondria is the place of respiration. It's the place where energy synthesis, like where energy produced. So suggest why the inner membrane of mitochondria is folded. In the question, they give us answer that it is used in respiration. So if it is used in respiration, so it means that it increased the surface area for the process of respiration. Different enzyme that used in the process of respiration, it work here, so it increased the surface area for that process of respiration, and we know process of respiration is the process through which any organism produce energy, right? So if more it's folded in more surface area, it means more enzymatic work, it means more like more area for the process of respiration. Large surface area, so more surface for respiration. Okay. All right. Now let's move toward another question. Table 1.1 show different specialized cells and the average number of mitochondria each cell contain, right? Specialized cells that given to me, it's liver cell, red blood cell, sperm cell, and heart cell, right? Heart muscle cell. Now, mitochondria is the powerhouse of cell. It means the energy production here occur in mitochondria. It means the energy that is ATP produced in mitochondria. So liver cell, if you see here, the number of mitochondria is more, while the red blood cell, no mitochondria, while the sperm cell, there is mitochondria, but relatively it is less than others, and heart muscle cell. Now take these two first of all, liver cell and heart muscle cell. These two need more energy. Why? The first thing is liver is the site of metabolic reactions. Most of the metabolic reaction happen here in the liver. Second, liver is the site of bile production. So for all these, energy, mitochondria should be more for the production of energy. While heart muscle cell is doing contraction and relaxation, and for contraction of the muscle we need energy. That's why the number of mitochondria is more here. Sperm, now come toward the sperm. Sperm cell need mitochondria to swim, but relatively less than heart muscle and liver cell. Why? Because sperm cell is small is compared to heart muscle and liver cell. Okay. Come toward the red blood cell. Red blood cell, it is not such active to move just like sperm and heart muscle cell. So it need energy, but very less energy. So and the like red blood cell should have more space there. That's why mitochondria is not there. So that the red blood cell should have more space for hemoglobin. So mitochondria is not there. So it does not mean that red blood cell no need of energy. Red blood cell need energy, but there is another mechanism happen only a very little, just like anaerobic respiration, a very little bit energy produced through that. Okay, I think it's clear. Now, if you want to take some notes from that, you can. Mitochondria is the site of aerobic respiration. Liver cell is very active. Sperm cell are active. They swim, right? All right, sperm cell have few mitochondria, is they are small. Red blood cell full of hemoglobin. Red blood cell use less energy, so red blood cell don't actually move, is like just sperm cell and like other like heart muscle cell contract, not like that. Okay. Now come toward another question. All right. Reflexes are simple responses that protect the body. The pathway that nerve impulse travel along during a reflex is called a reflex arc. It means the pathway through which nerve impulse travel during reflex action, that is called reflex arc. Okay, so little a to G show the component of the reflex arc. So let me tell you what is reflex arc. After that, we will come to our question. You know, first of all, it should be stimulus, like change in environment. If it is internal stimuli or if it is internal stimuli, change is necessary. Like if it is in the change of temperature, or if it is like certain pressure on the baroreceptor or whatever, but there should be change. If it is internal, like glucose level increase or decrease, temperature of the body increase or decrease, all these are stimulus. After that, stimulus well accepted by receptors. The receptor may be neuron ending or certain cells. After that, there's like this receptor, when it receives the stimuli, like when it receives the stimuli, electric impulse, like impulse will generate, and that impulse will start traveling to the nerve, sensory neuron. And it will reach to the spinal cord, and spinal cord contain interneuron or relay neuron. Through the relay neuron, the message will go from sensory neuron to motor neuron. Now, motor neuron is carrying that impulse, motor neuron is carrying that electrochemical signal to the effector, right? Now, the effector may be muscle or it may be gland. It depends on the situation. If it is muscle, it will start contraction or relaxation, right? But it can be like, if it is gland will produce hormone. What, like at that time, what is the need for the body, that hormone will produce. Okay, so that, and the response well made, like for example, it is muscle, so your muscle will start contraction and relaxation according to the situation. So this is the response. Now I think it is easy for you, that now you can do this question. The first thing, the first option is already given to us, A. So mean the first should be stimulus, right? And after that, what will happen? Stimulus well received by receptor. So I will put it here, the receptor. And then that impulse will travel to sensory neuron. So I will put it here, sensory neuron, right? And then it will move to the relay neuron. So I will take the, where is the relay? Yeah, I will put it here, the relay. And after the relay neuron, it will travel through motor neuron. So I will search for the motor neuron. It's a motor neuron, right? And then it should be the effector. So with us, this one is effector. And the last one is response, that one. You can check. Is it clear to all? All right, now let's move toward another. So this is reflex arc, just like the previous slide, if you want to know. So receptor, sensory neuron, right? Interneuron, motor neuron, and effector. Okay. Once again, receptor, sensory neuron, interneuron, motor neuron, and effector. Hope it's clear. Now let's move toward another topic. Another question is related with impulse travel between neurons across a synapse, across this gap. How the impulse is traveling from one neuron to another neuron across this gap. The gap is called synapse. Describe how impulse travel across the synapse from one neuron to another. So this complete mechanism, you should know about it. The first thing is, like this is the neuron ending of presynaptic nerve, and this is postsynaptic nerve. First one, through which the nerve impulse, through which electric chemical signals is traveling. Okay, so this is what this is. This is this part, like this is presynaptic nerve and this is postsynaptic nerve. Okay, so what happened here? The first thing, when the impulse reached to the presynaptic nerve, special channels will open. That channels are called calcium channels. And this channel, and you see it here in this presynaptic nerve, we have this vesicle containing neurotransmitter. These are what? Vesicle containing neurotransmitter. What are these? These are vesicle containing neurotransmitter. So when the calcium channel open, so this vesicle containing neurotransmitter, it will move toward the membrane of this postsynaptic nerve. It will move toward the membrane of postsynaptic nerve. Sorry, it will move toward the presynaptic nerve, and it will fuse with this presynaptic nerve, and that will release the neurotransmitter to this gap. The neurotransmitter will release to this gap. Okay, and that neurotransmitter will travel, it will travel and will move toward, on the opposite side there is postsynaptic nerve, on the opposite side there is postsynaptic nerve. On the postsynaptic nerve there are what? There are receptors. Is it clear? So these are the receptor for what? For the neurotransmitter that released from presynaptic nerve. Now this neurotransmitter will travel and it will move toward that receptor. It is on the postsynaptic nerve. So when this neurotransmitter that are released, when it is here in the synapse, and now it will travel toward this receptor. So it will bind with this receptor. And with it bind with the postsynaptic nerve receptor, here a channels will open, right? That channel will open for sodium ion, and influx of sodium ion will happen. Influx of sodium ion means too much sodium will enter to the postsynaptic nerve, and it means this postsynaptic nerve is now depolarized. It means impulse is now start traveling through this postsynaptic nerve. Impulse is actually what? The flow of these ions. So now the sodium is inside the postsynaptic nerve. So this is the way through which impulse travel from one neuron to another neuron. Okay, action potential reach axon terminal. Action potential means impulse reach axon terminal. Calcium channels open. Calcium causes vesicle to release neurotransmitter. It causes the vesicle to fuse with the membrane of presynaptic nerve, and that will release the neurotransmitter. And neurotransmitter crosses the synapse, and then it will bind with the neuroreceptor and trigger the signal in postsynaptic nerve. Hope it's clear to all of you. If you want to take like impulse arrive to the axon terminal of presynaptic neuron, trigger vesicle containing neurotransmitter to fuse with the plasma membrane, neurotransmitter release into the synaptic cleft and diffusion across and bind to the receptor on postsynaptic nerve membrane. Trigger impulse to continue along. Okay? If you want to. If you want to study the slide, you can. All right, now let's move toward another question. All right. Okay, drugs such as heroin resist the moment of impulse across the synapse. User can become addicted to the heroin when users stop taking heroin. They may experience withdrawal symptoms, state 2 withdrawal symptoms that heroin user may experience. Second question suggests why heroin abuse may increase criminal activity. Okay, so here, there, like I will take some general symptom of that when a person like he's stop using heroin, withdrawal symptom that heroin user may experience, like, so what are these are general things I will sleeplessness because he is addicted of heroin, nausea, aggression, vomiting, and headache. These are the general symptoms for that. Suggest why heroin abuse may increase criminal activity. So it will increase criminal activity because, you know, like that person, well, he's right now, he's addicted of heroin and he need to buy a dose of that. And if he having no money, so he is turning to the crime. Is it clear? Okay. Now move toward another. State 2 withdraw. All right, it's finished. Come toward another. Heroin abuse may lead to HIV infection, and there is currently no approved vaccine that prevents the spread of HIV. Vaccination stimulates active immunity against specific pathogen. Right now, there is no such approved vaccine for the prevention or for the spread of HIV. Now, here a question is about the vaccination. The first thing we should know about the vaccination, like what is vaccination, what is vaccine, and how it stimulates active immunity against specific pathogen. So first of all, I will move toward vaccine, like I will move toward vaccine. I will discuss first of all what is vaccine, right? So you know, vaccine is actually like a part of like attenuated pathogen. Like vaccine is actually attenuated pathogen. Attenuated means whose pathogenicity is being decreased. Like it cannot by certain chemical treatment or by heat, it cannot cause any disease, right? Second, vaccine may be heat killed or it may be a antigenic part of a pathogen that extracted, that antigenic part extracted by scientist. And this part having the ability to stimulate our immune system. It means that our immune system having the ability to recognize that. Okay, so if you inject that to your body, it will do nothing with you, like it will not cause disease to you, but your immune system will stimulate and it will start production of again antibodies against that pathogen. It will start production of antibodies, specific antibodies against that pathogen before the actual pathogen. So it means your body is now, he know, like before the actual pathogen come, before that he know that he having antibody and he having the ability to protect himself directly if against if the real pathogen come to the body, right? So this is vaccine, right? In the process that we are administrating the vaccine, it's called vaccination. Now explain how vaccination stimulate active immunity, right? How it stimulate active immunity. So before discussing active immunity, I should know about the type of immunity. Look at here, like we have two types of immunity, active immunity and passive immunity, right? Acquired immunity, two types, active and passive immunity. Acquired immunity is actually immunity that developed during your lifetime. So we have two types of immunity, active and passive immunity. Active immunity develop in response to infection or vaccination. Active immunity, when it develop, when your body exposed to pathogen, when your body enter to, when pathogen enter to your body and your lifetime. So against that pathogen, antibodies and memory cell will develop in your body. Or vaccination. So vaccine, I told you, it is like harmless pathogen, or it is a part of pathogen, right? So that will again stimulate your immune system to produce antibodies. So we have two types of natural and artificial active immunity. Natural antibody develop in response to an infection, while artificial antibodies develop in response to a vaccination, like a response to vaccination. Is it clear? So here in both cases, your body, like your immune system will be stimulated. And when your body's immune system stimulated, so what will happen? B lymphocyte will divide and one B lymphocyte will become plasma, and that will start production of antibodies against that pathogen, while another will become memory cell, and the memory cell will remain for a long time in your body. If next time this pathogen come, that memory cell will divide and it will suddenly start production of antibodies without any like delay. That's why if second time infection of the same pathogen happen to you, you will recover it from that very easily, like very quickly, right? Now, passive immunity here develop, like your immunity develop after you receive antibodies from someone or somewhere else, like from someone you ready-made antibodies you receive. In this case, you are receiving ready-made antibodies from another person. While in active immunity, you are developed antibodies and memory cells are developed in your own body. While in this case, in passive immunity, antibodies you receive from mother. These antibodies develop naturally and you are receiving from mother. So it's called natural passive immunity, while artificial antibodies received from a medicine, like from a gamma globin injection or infusion, like gamma globulin injection. These are certain antibodies that develop in the lab and that are injected to our body. So these are artificial antibodies, right? Is it clear? Now let's move toward our question. I think our question that will be clear now. Explain how vaccination stimulate active immunity. So harmless, dead or weakened, attenuated pathogen injected to our body. Antigen and vaccine triggers antibody production by lymphocyte and memory cells are produced that provide long-term immunity. If you see here, like first of all, what happened with us, like T helpers here present the antigen to the B cell, means B lymphocyte, and it is T lymphocyte. So B lymphocyte, when it present the antigen to B lymphocyte will start mitotic division. Mitotic division means it will start mitosis, right? And when it start mitosis, so one cell will form memory cell while the another will be plasma cell. Plasma cell are the ectolymphocytic B lymphocytes that produce antibodies at that time, while this memory cell will remain for a long time in your body. If next time the pathogen come, this plasma memory cell will divide quickly and that will start production of antibodies. Is it clear? All right. Now let's move toward. All right. So explain what is meant by passive immunity, right? It's a short term, active immunity, I told you it will remain for a long time in your body because of memory cell production. While passive immunity, it will not remain for a long time in your body, okay? Because here we have no memory cell produced. So no antibodies, like short-term defense against pathogen, no immune response, like you are taking antibodies from another. So there is no any immune response and no antibodies produced by your own body, antibody acquired from elsewhere, right? So that's why that will not remain for a long time. Passive immunity will not remain for a long time in your body. Okay. Now let's move toward another question. Yeast is used in bread making and it respires anaerobically producing carbon dioxide. Write the balanced chemical equation for anaerobic respiration of yeast and bread making. So if anaerobic respiration, so it means respiration, it means the breakdown of glucose and the absence of oxygen, right? So write the balanced chemical equation for anaerobic respiration of yeast and bread making. So this is the anaerobic equation, like it's a glucose and that will produce ethanol plus carbon dioxide, right? If it is happening, and remember it here, it's important point. If this anaerobic respiration happening in human, remember the ethanol will not produce there, lactic acid will produce, and no carbon dioxide will produce in human, okay? A baker wants to increase the rate of carbon dioxide production in the bread making process. The baker trial different concentration of glucose solution in the bread dough. Figure 3.1 show the result. What he's doing is the baker is actually increasing the concentration of glucose. And is it increasing? The rate of carbon dioxide production is increasing because there is more substrate, because there is more glucose for that anaerobic respiration and more carbon dioxide releasing. Now, at the initial, the graph is going very stiffy and gradually it reached to constant again. Okay. Now, let's see what are the questions they are asking from us. First is predict the rate of carbon dioxide production if the concentration of the glucose was 7%. So if we go here, like it's like from here, if the graph become constant, so what will happen with us? So again, that value will give us like 4.1 value will give us, because here already the graph reached, it's now fully saturated, it's reached to the maximum. So like because a certain limiting factor, now the graph are what it is constant. So our value at 7 should be 4.1. Its same value will come with us. Second, the baker carry out the trial at 30 degrees Celsius. The trial was repeated at 20 degrees Celsius. Draw a line on the figure 3.1 to show the rate of carbon dioxide production in 20 degrees Celsius. Right? So here, if you look at here, it's 30 degrees Celsius and it is 20 degrees Celsius. We know like enzyme and substrate, like for enzyme, they need optimum temperature. So if you are increasing the temperature up to specific limit, the rate of reaction will increasing. So, but if you are decreasing, the enzyme activity will be affected and the rate of reaction will decrease, because I told you already that temperature increase the kinetic energy of molecules. So if it is more temperature, more collision between enzyme and substrate. But if it is less collision between enzyme and substrate and less chances of conversion of substrate to product. So it is 20 degrees C, it is less than 30 degrees C. So the graph will be same, but it will be down, it will reach to the maximum, but it will go like it will be down, it will be not at a very high speed, not like this steepy, it will not go, but it will be down from this. Okay. Now, the baker carry out another trial at 80 degrees Celsius. No carbon dioxide was released. Now the baker increased the temperature. And I told you there should be optimum temperature for the enzyme. If you are increasing above that, the enzyme will start denaturing, because enzymes are proteinaceous in nature. So here, the baker carried out another trial at 8 degrees Celsius. So it means at 80 degrees Celsius, it is more than like the optimum. By this temperature, no carbon dioxide release. Why no carbon dioxide? Because the enzyme were denatured. Okay. Name one another industrial process that uses yeast. So yeast can use and different processes, we can use, for example, for biofuel production, for ethanol production. All these are the uses of yeast. Okay, even we can also convert like yeast to yeast extract, genetic modified yeast. All these are the industrial processes that using yeast to fruit and industry. They are also produce genetic modified yeast for different purposes. If you want to note the answer, you can. All right. Now let's move toward, I think this question is clear to all. Let's move toward another question. During the production of penicillin, large fermenter are used. You know, like fermenter is a vessel where we are growing fungi or like bacteria so that it produce certain product. Where we are in fermenter, we are providing control environment to that organism, like pH, temperature, nutrient. All these things we are providing to the organism, and they are producing like what and which thing, like which we are interested in. For example, here they are interested in production of they are interested in production of penicillin. So the penicillin antibiotic, so for that definitely they will grow fungi in this vessels, right? But this vessel should be like controlled. So this is the water filled jacket of this fermenter. This is probe through which we are like water filled jacket. It's used to like to, if the temperature is, it should be, for example, if temperature is more, so we are like, oh, cool water is coming toward that. If temperature is decreasing, like it's actually absorb the heat from the fermenter, right? So that the temperature may not increase than normal, otherwise the fungal will not grow well. So this is the function of water filled jacket, right? So it's, this one is probe. It's like there is different probes, so different for probes having different function. Like some probe are detecting the pH, so that because we have to provide specific pH to the organism. Some are for the temperature, that record the temperature. Some probes are for even for the nutrients, like we are providing NH nutrients for the growth of organism. Then later on it will produce our product, right? And this, like you can see here, this fan like structure, these are stirrer. Like this stirrer help to homogenize the mixture, like so that all the nutrients, there should be nutrients should circulate in this whole system, so that there should be more supply of that nutrients to the organism, right? And so now let's see the question about this fermenter. What are the question with this? Explain what is the function of the following part of fermenter: stirrer, water filled jacket, probe. Okay, so the stirrer, I told you, keep microorganisms suspended, prevent it from sinking, enable microorganisms to always have access to nutrient, and maintain even temperature. It is another point, to create uniform homogeneous mixture. Okay, now water filled jacket. Reduce heat energy, like it's absorbing the heat from that. There is a constant supply of cool water. So if heat is produced as a result of metabolic reactions inside the fermenter, so that are absorbing that. Maintain a constant or optimum pH for that organism. Optimum temperature for that organism. Probes, monitor pH and temperature and nutrients, right? The air and nutrients that are added to the fermenter are sterile. State why they must be sterile. So why they must be sterile means pathogen free, so that it should be sterile. If we are not adding sterile air or nutrients to the fermenter, so it will cause contamination and contamination is the growth of unwanted organism in our fermenter instead of the organism in which we are interested. Instead of that, other organism will start growth, and that will disturb the whole system. So it means that why we are doing this, to sterilize it, we have to sterilize it in order to prevent contamination. Is it clear? All right. Okay, now question number 4. Increased human population is linked to change in carbon dioxide concentration in atmosphere. Figure 4.1 show the carbon dioxide concentration between 1958 and 2012 measured at Mauna Loa, Hawaii, right? So like in different year, if you see like 1960 and that graph is going up and like it's steeply going up, and it's 2010, it's more like the carbon dioxide concentration is compared to 1960, it's gradually increasing, and now it is at a peak at 2010. Like if you are describing the graph, so first you have to see the general trend in the graph. What are they? Generally, there is decrease in the graph. Generally there is not increase in the graph. Generally there is carbon dioxide concentration is increasing. There is a little bit fluctuation between the years, right, or between different seasons. There is a little bit fluctuation, but overall our graph are increasing. And from the graph, if they want to describe you will do like this general trend in the graph, and after that you can give value from the graph like 2010. What should be the value? You will come and will give the value that is coming against it with carbon dioxide concentration parts per million. You should be careful about the unit. Whenever you are describing the graph, you have to mention the unit. Unit is very, very important with us, right? Describe how the carbon dioxide concentration has changed between 1958 and 2012. You will gain credit for using the data from figure 4.1. I discussed with you all overall carbon dioxide concentration increasing at a steady rate, and there is a minor fluctuation in carbon dioxide concentration yearly. Now, this explanation you will give you overall there is increase in carbon dioxide, but there is a minor fluctuation up and down is there, right? Now, after that, you will give the data from the graph with the unit, okay? This is how you will describe the graph. When your graph come to you, so you have to be first give the first sentence that what is the general trend it's happening in the graph, like it is increasing or decreasing, or it is increasing, or if it is increasing, how it is increasing, like it is steeply increasing, gradually increasing, steady increasing, like it's become constant, and how it become where it decrease. All these points you have to mention whenever you are describing the graph, right? All right, now let's move toward another. Carbon dioxide is a greenhouse gas. Now name one other greenhouse gas, right? Greenhouse gas, I like greenhouse effect. Last day I discussed with you what happening in greenhouse, right? So just here there is like different greenhouse gases that cause greenhouse effect, but the one gas I told you, like carbon dioxide, methane, chlorofluorocarbon, all these are nitrous oxide, all these are greenhouse gases, and that make a blanket like this, right, around the like in the atmosphere. And I explained this actually already, right? So let me tell you in detail about it. You know, like once again I will just repeat to you again. When the sunlight is coming toward, like explain how carbon dioxide enhance the greenhouse effect. So how it is enhancing the greenhouse effect, like when the sunlight is coming toward the earth surface, right? So it's short, the like the wavelength is short. If the wavelength is short, penetration power is more, so they can pass through this blanket of greenhouse gases. When it struck the Earth's surface, it will lose energy, and the wavelength will become like long, right? Now, some of the rays absorbed by the Earth's surface, where some will go back, and the wavelength will become long. And when its wavelength become long, its penetration power is not high, and that cannot penetrate through this layer of gases. It cannot go back through this, and that remain in our atmosphere here near to the surface of the earth, and that lead to global warming, right? And this is called what? Greenhouse effect. You can study, please. Is it clear? All right. Now, let me tell you about it. Then I will ask a question from you if you're having any doubt. So minerals iron are needed for plant growth. Complete table 4.1 to show the function and effect of the lack of some of minerals iron on plants. Minerals that given to me is nitrate, magnesium, phosphate, right? So the one phosphate it is given to me used for the making DNA, right? If it is like no more in your in plants poor root growth. DNA contain, like the basic unit of DNA is nucleotide. If you study the structure of nucleotide, that consists of ribose sugar, nitrogenous base, and phosphate group. Okay, so phosphate need for the DNA production formation, while nitrate and magnesium forward. So we know, like if we study the structure of protein and especially the structure of amino acid, they need nitrogen, and that come from nitrate. So what is the function of that? It's actually make amino acid, protein, DNA, RNA, and all these, this nitrogen, nitrate play important role because it is providing nitrogen for all these structures. In all these structures, the nitrate is very essential. If it is less in the plant, so poor growth, lower leaf, right? Lower leaf died early, right? All right, now, second one, we have magnesium. Magnesium used to make chlorophyll. It is need for the plant chlorophyll, and we know chlorophyll is the pigment in chloroplast. It's necessary for the process of photosynthesis. If there is less chlorophyll, the yellow leaf will become yellow and chlorosis will happen. Now, I will not move to this topic now. In next lecture, I will proceed toward the eutrophication, and that now I will discuss with you that like if you're having any doubt, any question, you can ask from me. Okay, you can raise your hand. I will take one by one all of you. Let me connect my. All right, please.